Carbohydrates are essential components of all living organisms and the most abundant class of biological molecules. The name carbohydrate literally means carbon hydrate, stems from the chemical composition, which is carbon, hydrogen, and oxygen. The basic units of carbohydrates are known as monosaccharides. Oligosaccharides consist of a few covalently linked monosaccharide units, whereas polysaccharides consist of many covalently linked monosaccharide units. Here you can see several common types of carbohydrates. Starch is a major plant polysaccharide for storage. Corn, potatoes, and bread all are rich sources of starch. Animal cells store carbohydrates in form of a polysaccharide called glycogen. In this figure, glycogen is stained purple in these animal cells. Honey is a commonly used product and is a mixture of various mono and oligosaccharides. Carbohydrates also have a structural role. For example, plant cells contain cellulose in the cell walls. In this electron micrograph, the cellulose fibers in cell wall are visible. The exoskeleton of insects is made up of chitin, another structural polysaccharide. Nucleic acids, DNA and RNA also contain a sugar molecule as a component of constituent nucleotides. This figure is a representation of DNA structure in which deoxyribose sugar is present. With this background, let us now take a bit more detailed look at the roles of carbohydrates. This section provides a broad overview of some important functions of carbohydrates and is for your information only. All of this information is not present consolidated in one book chapter. You are not required to remember the structures shown in this section. Carbohydrates are a very important source of energy. Living things store carbohydrates in form of polysaccharides, which can be converted to monosaccharides when needed. Glucose is the major monosaccharide to produce energy, ATP, through oxidative phosphorylation. Several of the intermediates of carbohydrate metabolism are important for other processes taking place in a living organism. For example, 1,3-bisphosphoglycerate 1,3-BPG is an intermediate of glycolysis. Another related derivative 2,3-bisphosphoglycerate 2,3-BPG is an important regulator of oxygen binding to hemoglobin in red blood cells. Acetyl-CoA is a 2-carbon donor compound which feeds carbons to Krebs cycle. Moreover, acetyl-CoA is also a precursor for fatty acid and thus lipid biosynthesis. In this structure, acetyl group is shown in red color. Rest of the molecule is coenzyme A. Oxaloacetate and alpha-ketoglutarate are also intermediates of Krebs cycle. In addition, these are precursors to amino acids aspartic acid and glutamic acid, respectively. The structures are shown in the figure. Succinyl-CoA, another intermediate of Krebs cycle. Its structure is shown here. The 4 carbons shown make the succinyl part of the molecule. Structure of coenzyme A is not shown but is abbreviated as CoA. In addition to its role as a Krebs cycle intermediate, it is also a precursor for heme biosynthesis. Carbohydrates are also important for cell adhesion. Cells are attached to the extracellular matrix, which is a complex made up of several different types of proteins and carbohydrates. specific types of carbohydrates are present on cell surface and they attach cells to each other. Another example is leukocyte rolling. Leukocytes are white blood cells. They are important for immune function. Normally they float in bloodstream. But when they sense an infectious agent in the surrounding tissue, they attach to capillary wall and start rolling. In this microscopy video of a real capillary, you can clearly see a leukocyte rolling along the capillary wall. This rolling is mediated by specific carbohydrates present on cell surface. The leukocytes eventually squeeze themselves through the inner capillary wall and migrate to neighboring affected tissue. These cells are shown by 3 red circles below the capillary. Carbohydrates and the derivatives are also important for cell signaling and recognition. For example, inositol is a reduced derivative of glucose 6-phosphate. Phosphorylated inositol is a component of polar head group of membrane phospholipids. As a result of certain signaling stimuli, inositol triphosphate is released from the membrane into cytoplasm of the cell and mediates signaling. ABO blood groups are different from each other because of the different carbohydrate part present in them. Blood group antigens are present on RBC membrane and are important for blood group compatibility during transfusion. Carbohydrates are attached to many proteins, a process called glycosylation when they pass through the Golgi pathway. These proteins are called as glycoproteins. Many glycoproteins and other related molecules are present on surface of cells and act as an attachment site or recognition molecule.